Professor Mart Saarma, counting Viking nations and academies of sciences. I forgot to mention that Ivan is member of 2 academies, Royal Society and medical sciences. But Mart Saarma is member of 3 Viking nation academies of sciences, Estonia, Finland, and now also Royal Swedish Academy of Sciences, awarding Nobel Prizes. I got to know Mart's name long before Juvan's name. I was PhD student in Helsinki, and Mart was already big name at that time. So I believe from 1990 already heading the Department of Biotechnology at University of Helsinki. So I was studying algorithms, and I did not know anything about biology at that time. But Mart has been a very big name in Finland, in Finnish research community, and associated most of it is Institute of Biotechnology at University of Helsinki. And several prizes, etc. But let the man speak on his own behalf. Mart, please, the floor is yours.
Thank you. Thank you, Jaak. And what should I say? I have certain advantage compared to Ivan, and that is that I have many friends here. I have even my former PhD supervisor here. And then I have classmates and some PhD students as well. So a few years ago, when my oldest grandchild went to school, she one day came to me and asked, "Grandfather, tell me what you are doing." And I tried with my best capabilities to explain her what I'm doing. And next time when I met her, I realized that at school, when she was asked what I'm doing, she was proudly announcing that my grandfather is a mouse doctor, hiirearst. So here I am, hiirearst. So I thought I will thank you. I will talk you today about the life and death in the nervous system. I will try to tell you that the cells, what you are using for thinking and memory and for loving and expressing feelings, are the most beautiful cells in our body. And I will first introduce you to the neurons, and then discuss with you how do they die, and why do they die. And actually, from the point of view of therapy, it's very, very important to know, can we prevent that death? So you see here on the right a nerve cell. And actually, the nerve cells have, I would say, 3 major important functions. First, to receive information. Then second, to integrate and store it, and then to transmit it. And that is actually occurring through a special organelle which is called synapse. And you might know that in our brain we have altogether 10 in the power of 11 synapses, which actually is neurons, which make altogether 10 in the power of 14 synapses. This is actually almost 500 times more than we have stars in the Milky Way galaxy. So, single neuron, and I will talk today these champions, and these are dopamine neurons, can make synapses with 500,000 other neurons in the brain. That means that every one citizen in city of Tallinn would be able to talk to every citizen in city of Tallinn, send information, store it, and receive. Quite a challenge. So the human brain is roughly 1.5 kilos. If we compare us with a chimpanzee, that is about 700 grams. Whales and also dolphins have bigger brains, but they have less contacts. So although the brain is half of the size and the number of neurons in between chimpanzee and human is the difference is only 3 times, the number of synaptic contacts is 10 times different. And if we go to the cortex, the area which is really important for the memory and learning, the difference is 100 times. So actually, the synaptic contacts are the major players. So via synapses, neurons communicate and form neuronal networks. And what is probably very, very important, especially for this audience who are working with computers, is that our brain and synaptic contacts can be modulated. And actually, what is even more important, the so-called plasticity of the brain, or the ability to memorize and think, is based on the brain ability to finish some of the synaptic contacts and form new contacts. So this is actually, as you can see, 10 in the power of 11 neuronal contacts. This is endless. This is really endless. So once again, the memory and learning is based on the ability of neurons to modulate synaptic contacts and to get rid of old ones and form new ones. What is also very important is that most of the nerve cells in my brain is actually as old as I am. And the same in your brains. Only a tiny, tiny, tiny fraction of the brain neurons can be renewed. Well, these are important. Also, some of the neurons which are renewed are very crucial for learning new things. But by and large, they are not renewed. So there are 2 choices. They either stay alive or they die. And that's really what happens quite massively. And about the life and death of the neurons, I will talk today. To understand the complexity of the system, I have on the right side shown you also a picture of one single dopaminergic neurons. These are neurons which are responsible for most of our motor coordination. This is actually a picture of the single neuron, which has only one cell body. And this cell body gives rise to the axons which are branching in the mouse brain 100,000 times and make 100,000 synaptic contacts. And in my brain and your brain, 500,000 times. Imagine that this cell body has to transport molecules and energy to 500,000 different locations. That is why also brain uses 20% of our body energy. It's really a hot place. So, as I said, neurons form neural networks. And as we know from the very recent Nobel Prize winner's work, Moser's work, so these neuronal networks really play a very, very important role in how we recognize a space, how we see, how we hear. And the important point is that the contacts between these very simple 4-neuron network are changing. Now, neuron 1 fires to neuron 2, neuron 2 to 3. But in the processes, memory and learning, the synapse between neuron 1 and 2 disappears and the new forms between neuron 1 and 3. And that is an elementary event of learning. So neurons die in development, they die in aging, and they die also in disease. So very roughly in the brain in my brain and in the brain of a graduate student is already a 20% difference. I have roughly 20% less brain neurons. They are gone, and they are gone forever. And this process is unfortunately continuing. I don't know how long, but let's hope at least to the end of this lecture. So I found actually a quite smart pearl from Buddhist wisdom, which tells, and very correctly tells, that life is uncertain, death is certain. And that is really true, and that is also true for neurons. So how we currently view on the cell death, which is an elementary process of life. Broadly speaking, there are two classes of death. One is accidental cell death, very often also called necrosis. And this is a bad way of dying. The cell membrane will be broken, everything falls out, and inflammation is a consequence. And then we have different strictly genetically regulated cell deaths, one of which is apoptosis, which many of you probably have known, but there are many others like necroptosis or pyroptosis. And the main importance of this genetically controlled cell death is that first, the cell membrane will not be broken, and all the materials which are in these cells are reused. It's a dream of our contemporary environmental policy. Our cells know that already for a long time, how to do that. This actually occurs quite massively in the early childhood. Everyone who has had a child remembers that the children have a big head, unproportionally big head. And that is because they have 2 times more neurons in their brain. And then, shortly after the birth in the brain, in different areas of the brain, these neurons start to die. And there is a massive neuronal death in the brain of the child during the first year of life. The biological meaning of that is to guarantee precise synaptic formation. We call it wiring. And many of these contacts will stay forever. And then during this 1 year, half of the brain neurons are brutally killed. But they are killed by this apoptotic, clean, genetically controlled system. So we call it programmed cell death. There are butchers and there are warriors who help to rescue cells. Caspases are the major enzymes which are the major butchers. And we know nowadays quite a lot about how they function. We have different pathways. We know quite well them as well. We have, in some cases, shown on the left with the green, that you have certain proteins which bind to the receptors and induce cell death. And then we have a mechanism which involves mitochondria and renders to the cell death. We now know quite well about these. We can also model neuronal cell death in the test tube. So we can grow neurons and induce different types of cell death. And we can keep them also alive. Nowadays we can also grow human brain cells, thanks to the stem cell technology. I am currently growing in the lab human dopaminergic neurons. And they actually look very much like normal dopaminergic neurons. They have the spiking activity, they produce dopamine, etc. So we can even build mini brains nowadays in the lab. So we have molecules which kill neurons, but we have also molecules which rescue neurons. And the best characterized proteins are called neurotrophic factors. These are factors which really keep neurons alive. They were discovered in the mid-50s. Actually, the story is very interesting. They were discovered by an Italian lady, Rita Levi-Montalcini. She was born in a Jewish family in Turin, in Italy, and she was not allowed to work during Mussolini time at the university. But she was from a rich family, so she built up a lab in the kitchen. And the first experiments on the discovery of neurotrophic factor, Rita did in her kitchen, which is quite remarkable. So, few years ago, she and then her biochemist student Stanley Cohen got the Nobel Prize. And I put also Victor Hamburger's picture here because actually the final work was done in Wash U in St. Louis in Victor Hamburger's lab. And Victor actually in early 30s predicted that these growth factors exist. I show that picture also for the other reason, and if you look carefully, you see that Stanley died at the age of almost 100. Rita, as a member of the Italian Senate at the age of 104. Victor Hamburger at 102. So isn't there a correlation between what you investigate and the continuation of life? So I thought I look also quite good. So because I want to tell you today about the very specific set of neurons, and these are the dopamine neurons, and these are the neurons which I said are regulating our motor coordination, or how can I raise my hand, can I move my leg. I will focus on also two growth factors which are very important for these type of neurons. And these are growth factors of the GDNF family. There are actually 4 members of the family. They are small molecules, about 100 amino acids long or a little bit longer, and they form homodimers. 2 identical subunits make a homodimer. And this homodimer then how can I show? Do you see? This one. Okay, sorry. So this is a GDNF. This is a core receptor. So the ligand binds on the cell membrane first to the core receptor, and then there is a transmembrane protein which transmits signal into the cells. So the growth factor first binds to the core receptor, then core receptors form a complex with transmembrane receptor, and without entering into the cells, they put these 2 receptors together and activate. In this particular case, actually, phosphorylation of the tyrosine residues in the cytoplasmic domain. And these tyrosine residues then serve as docking sites for the activation of intracellular processes. We activate rapid processes, but we also activate transcription factors. So actually, if you treat your cells one day with a growth factor, they will remember that for several months because you activate growth factors. It's a long-lasting process. And then, of course, we know exactly where GDNF binds. We know exactly what are the tyrosines they activate. If we mutate one single tyrosine in the cytoplasmic domain, receptor is completely kaput, can't do anything. So we know quite well how it works. So many, many years ago, we actually, my lab was the one who also discovered the GDNF receptors, and later we then did the structure, and I come back to the structure. Here you see that how these homodimers bring together two core receptors and then form the big complex. So quite interesting, a few years ago, several labs, including my lab, discovered that one of the growth factors called GDF15, and that's actually a very hot molecule. GDF15 is a growth factor which strictly regulates the appetite. And in mice, if you inject that growth factor, in 1 week, the body weight drops 10%. So pharmaceutical companies are now running after that molecule. And 4 labs at the same time actually showed that this new growth factor actually also signals through the same ret as GDNF. So it's the same family. We suddenly started to investigate also the body weight control. Just was not planned, but so it happened. So as I said, neurons die not only in aging, they also not die only in development, but they also die in diseases. And one of the bad diseases where neurons die is a disease called Parkinson's disease. In this disease, the dopamine-producing neurons, which are located in the substantia nigra, they send their axons to the caudate putamen. These are the neurons which make 500,000 synapses per neuron. And these are neurons which produce dopamine. They are actually a dopamine-producing factory. And then they release dopamine in this caudate putamen. And in the Parkinson's disease, these neurons die. And actually, this is a quite amazing example how plastic we are. The patient has lost 50% of the dopamine neurons in the substantia nigra and still has no symptoms. And then he or she loses another 5%, and then she or he starts to get the motor symptom. Until 50% is not lost, we have very strong compensatory mechanisms. So I want to show that slide because we all thought that Parkinson's disease was first diagnosed by a British doctor and mineralogist, Dr. James Parkinson, but if you read the literature, you can find out that the disease was actually described in China and India much, much earlier. And if you look the Ayurveda, the life of science of life, then they describe the disease under the name Kampavada. And actually the description of the disease is 100% the same. But what is much more amazing is that at that time they also proposed treatment. And for the treatment, there were 3 recipes. And all 3 recipes contained the extracts from the plant Mucura pruriens, that's a common bean in India, and the seeds of that plant contain levodopa. And levodopa is actually what is used for the treatment of Parkinson's disease, and the Swedish scientist got the Nobel Prize for that. Well, was known much, much before. Actually, the 2 other recipes are very interesting. There are some other very interesting ingredients which have not been very carefully studied, but maybe very important. When I gave a lecture at the University College London and showed that slide, science historians raised hand and said that, no, this is not James Parkinson's you are talking about, because photography was invented 100 years later. This is a stomatologist, Parkinson's. So I don't know whose picture I'm showing to you. Maybe the right one, maybe the wrong one. So when you look at the brain of the Parkinson's patient, then in a very, very simplified way, the picture, what you see at the moment when the patient gets a diagnosis, is as follows. So you have still maybe 50% of the neurons which are okay. They make synapses, they produce dopamine, but eventually they die. I will show you soon a picture, you see how quickly it happens. And then we have maybe 10-15% of the neurons which have lost synaptic contact, but the cell is still alive. And then we have maybe 10% of the cells which are still alive. They can be stained with neuronal markers, but they are not able to make dopamine. And then we have dead cells. Well, dead is dead. There is nothing to do. But these three groups of cells are actually what we are trying to rescue. We try to rescue these because they will eventually die, and these we try to regenerate. So current therapies, what we have, all of them, not only for Parkinson's, but all neurological diseases, are symptomatic. None of the treatments would slow down or stop disease progression. So what we want to do, what future therapies should do, is to try to prevent the degeneration of these neurons and perhaps have a treatment which can regenerate them. So this is a little bit sad to read, but there are many limits in the current knowledge of Parkinson's disease, and that is true also for Alzheimer's or Huntington's or ALS. We lack early diagnosis. We don't have biomarkers. We do not understand the pathogenesis, although for 20 years we know about 24 of the genes which are responsible for the familial forms of Parkinson's. But still, we are very much in the black box. And then we don't have drugs. So what I wanted to show you is that, and underline that, on how many planets Homo sapiens has been? Just Moon. How many guys died, starting from Michelangelo, before Armstrong went to Moon? I guess hundreds, maybe thousands. And remember, Milky Way galaxy was a piece of cake compared to our brain. So these are very complicated systems. So it's maybe also not so realistic to expect very quick breakthroughs in very short of time. So I show you again this dopamine neurons. We have 500,000 dopamine neurons in both parts of the brain, and each neuron can make 500,000 synaptic contacts. It's a complicated system. So we know something on here. You see the post-mortem picture of the brain. This is the color shows not essentially the neurons, but their axons. And you see here healthy brain, 1 year, 3 years, 4 years, 5 years, 11 years. You don't need to be a neuroanatomist to see that actually in 4 years, the game is almost over. There are no fibers anymore. So we have to start to treat earlier. That is very, very important. And then I think another very important thing is that we have in these dopamine neurons Lewy bodies. Lewy bodies is interesting. Many of you know that Emil Kraepelin, the father of psychopharmacology, worked at the University of Tartu. Benjamin Lewy was Kraepelin's student. Neurons are the most common staining for neurons is Nissl staining. Nissl was Kraepelin's student. You know all Alzheimer. Alzheimer was Kraepelin's student. So it was all started from Tartu. Quite important. So these alpha-synuclein, which is a major component of the Lewy bodies, is a hallmark of dopamine neurons in Parkinson's disease. And what we know now is that this is a prion-type protein, which we don't know exactly how it gets into the brain, but it gets misfolded. And when he meets the healthy alpha-synuclein in healthy cell, it gives a killing kiss. And the cellular alpha-synuclein becomes also pathologic and start to spread. And a lot of hopes are now around this alpha-synuclein. So about this growth factor, GDNF has been in animal models and also in clinical trials. And because it is a protein, it doesn't pass, there is a blood-brain barrier protecting our brain from toxic substances and others. But the drugs, what you want to treat brain, have to pass that, and proteins don't, so you have to put the proteins directly into the brain. So what we do, we inject the protein either as a protein or as a gene therapy vector or as a cell vector. And in animal models of Parkinson's disease, this GDNF and the related protein actually protect and repair dopamine neurons. There are some caveats, like they don't diffuse well, they don't work on all models, but they actually work quite well. So encouraged by these experiments on rodents and also non-human primates, GDNF was taken to the clinical trials. And GDNF protein has been in 3 phase 2 clinical trials. Phase 2 clinical trial means that you have also a placebo group and then the group who gets the drug. Well, the first 2 trials failed, largely because they were not well planned. This is not a blaming because it was a new area. We didn't know how to plan. In this very first one, they injected the protein into the cavity, what we have in the brain, into the ventricle, and it just didn't diffuse to the dopamine neurons. We know that very well. But now, the third trial, which was conducted in Bristol by Steve Gill, I was actually sitting at the advisory board of that trial, showed first time actually, in my opinion, a great promise. So the GDNF was delivered using these 3 catheters, which are inserted into the putamen, and then to this place an outlet which combines all these 4 channels are brought. So the patient comes to the clinic, is connected to the pump, and during 2 hours the drug is pumped to the brain and it diffuses to the brain. So these results were very, very encouraging. First, almost half of the patients had significant improvement in the motor behavior. This is definitely the first time in the history of Parkinson's disease that we can show the slowing down of the disease progression. Second, we also using the PET and fluoride-labeled dopamine transporter agonists, that there is a increase in the activity of the dopamine neurons. And then when we look, these are old people, some of them have died, and we see from the autopsy that this is a before GDNF, a control, and this is, you see, really increase in the number of dopamine neurons. And in here, the upper shows before and lower the positron emission tomography. You can see the red label shows there are more neurons. But what is more, if we look at that picture, could you switch this? I have pictures of 3 patients before and end of the trial. And I think this is really remarkable. This guy is almost running. So I think that the therapy really works, but there are many open questions. So why only in half of the patients? And then you have to have the brain surgery, which is also complicated. So what can we do better? First, currently, because it includes the brain surgery, the ethical committees do not allow to treat early stage patients. You can start from 5, 6, 7 years. I showed you the picture. Not much is left. Not much is to rescue. But if you could have some treatment avoiding brain surgery, you can start earlier. The GDNF drug is not diffusing very well. It doesn't cover the whole area where dopamine neurons are. And then Parkinson's patients, in addition to these motor symptoms, have also quite many non-motor symptoms. And some of them are actually very significantly reducing the quality of life of the patients. And these are constipation, depression, sleep disorders, etc. So what is a solution? Solution is to develop growth factor mimetics, small molecules, which would have the properties which protein has, can activate the receptor, but then has much better pharmacokinetic So we actually, together with Matti Karelson, who is sitting here in the audience, have started to look at that quite many years ago. And perhaps the intellectual starting point was when we solved the crystal structure of the GDNF and its complex with a receptor. And if you look at this ring, what I have drawn, there are 3 contacts of the ligand to the receptor, which are in the very narrow area. And each of these contacts, when we mutate it, destroys the interaction, telling that key interaction between the ligand and the receptor are in the narrow space. So, in principle, you can develop small molecules. So we went off to do that, and we did that in close collaboration with Matti Karelson. So we hope that the mimetics can cross the blood-brain barrier, spread well in the tissues, and treat also non-motor symptoms, and otherwise work like the protein. So this is a scheme how we worked. I don't bore you with that. But we started by using computational prediction. That was Matti's work. And we were actually lucky. We found a couple of dozen of compounds immediately which activated the receptor. But unfortunately, most of them were highly toxic. So when we injected them to the mice lost very quickly the hair and everything. So we had to look at other compounds. So we went then to develop a high-throughput screening assay, and then using big chemical libraries, we screened compounds. And to cut a long story short, we got one compound. Again, this was a collaboration with Matti Karelson, BT13, which actually fulfilled the criteria of the GDNF mimetics. You can see here that, like GDNF, it can activate red phosphorylation. It can also rescue dopamine neurons. And if we use a CRISPR-Cas and remove the receptor, it doesn't work anymore, so we have also genetic proof. And then we show that it works also in animal models of Parkinson's disease. But although the compound kind of work, we had the proof of concept, there were problems with this. They had poor solubility, they were very quickly degraded, they had somewhat high molecular weight, and maybe also not sufficient affinity to the receptors. So we have started about half a year ago a collaboration with a French company, Argo Bio, and in a relatively short period of time we have been able to improve the properties of this compound almost 100-fold. So I'm looking very much into the future in a hope that in a year or so we could start to take these new compounds into the clinic. And I hope they will work and eventually also help people who need their treatment. I will stop here, thank my colleagues and collaborators, and particularly grateful to Matti Karelson, together with whom I started this interesting journey. Thank you.
Thank you very, very much. I want to ask questions. So now the science is speeding up a lot. Now you would not have had to wait for crystal structure because deep learning could have helped you get some of the structures. 20 years ago,
you're talking about AlphaFold.
Yep.
AlphaFold is not predicting well transmembrane proteins.
Okay.
Sorry.
You can have arguments with Juvan?
Probably won't do the receptor ligand pocket.
So 20 years ago, you could have intimidated us with 500,000 times 500,000, and it would have been a big number. Now we know that computers are getting to the scale that we can start having computers or clusters that can match the human brain with the number of elements, connections. Deep learning is achieving a lot of training. What I want to ask you is intelligence, human intelligence. Many things that you spoke in here are kind of quite mechanistic approach to the brain, neurons, specific type of brain, neurons. What about artificial intelligence? What are we missing compared to your understanding of brain? Big jump to artificial intelligence. What is really this intelligence jump?
In my view, the fundamental difference is that in the computer, the connections are fixed.
Why? We can change them.
Based on the information which comes from outside.
Always.
So you can
programs can change things. We can do.
But you need a program. But you need a program.
Oh.
In the brain, under the influence of the information which comes.
Programs can be made self-evolving as well. So I'm provoking. What is missing from our understanding, from your point of view?
Well, I think in routine, computers, if the rules are sort of fixed, like chess, you do better. But if in the middle of the chess play, I will change the rules and start to play, I don't know how it is in English, vedelat. How do you manage? I think that was a
reinforcement learning.
I think
so
that
you start from scratch. You change the rules. You get different optimization methods.
I have seen a movie where the NBA basketball player and the computer were throwing free shots. And in the hall, without wind, the computer was 98 of 100. But then they started to blow wind from left and then change it to right. And the computer missed practically everything. So that is missing.
All the smart ammunition of nowadays is not guided by humans, but by computers. But let's not go into there. So,
if I may, Mart, I'd like to congratulate, because it's coming from the medical field and particularly in the Parkinson's disease and surgical treatments. I'd like to congratulate because this is something absolutely new. Because I know that you've been involved since early 90s, and I was aware about the techniques which was used in mid-80s. There was a really very big hope, but basically failed. But I think that your approach is now something which is completely new. So that once more, congratulations, and I think that all the surgical field is really proud of your achievement.
Thank you.
there was no question. Thank you.
Thank you, Professor Saarma, for this fundamentally interesting topic. I've had a fundamental question for 2 years already related to synapsin. I'm not investigating synapsin myself, but as it's a phosphoprotein and it forms highly condensated so-called phase condensates in the postsynaptic neurons, somehow probably governing the vesicle formation or vesicle transportation. Now, in Lewy bodies, we can see that actually the Lewy bodies contain several proteins. Major of it is, of course, the synapsin, and it contains also the remnants of the vesicles. Now, for me, there has been a fundamental question related to the issue of neurodegenerative disease and the connection between the synapsin structure. The question is whether the loss of synapsin structure is the consequence, or is it a cause of some kind of molecular disease-causing mechanism? Of course, loss of the function of the vesicles governing proteins cause the disease. That's clear. But what's causing the loss of structure of the governing protein? That is the question. And I have an experiment. I don't know if it can be conducted or if it has been conducted. There is possible to make a triple knockout mice in synapsin. The mice lives. Now there is also Parkinson disease model mice. Now if these two combined would rescue the Parkinson symptoms.
Yeah, you touch a very, very important question about, it's not only Parkinson's, it's in all neurodegenerative diseases, the troubles start from the synapse. This is the first event what happens. So I think that is one very good example, and that comes actually from the Parkinson's disease. I was talking about the alpha-synuclein, which is part of the Lewy bodies. So alpha-synuclein is a protein which is in all cells of our body. And in the nerve cells, it is part of the so-called SNAR complex. SNAR complex is actually a complex which packs vesicles which contain in dopamine neurons dopamine. And then alpha-synuclein is responsible that this vesicle is recognizing the cell membrane and facilitates its release. So now when you take alpha-synuclein away from the SNAR complex and put into the Lewy bodies, you have to have some troubles with the neurotransmitter release. And that's what really happens. So I would anticipate that many other proteins involved in the docking release of synaptic vesicles have significant impact to the loss of synapses and also through that to the neurodegeneration. I'm sorry, I can't give you a better answer.
We have a next question in here, Madis.
Thanks, and thank you for the lecture. My question is about the deep brain stimulation with electricity. So this is a technique that also requires invasive surgery, but how does it compare to this growth factor approach? What does it do to these brain cells? Does it just kind of mimics them or kills them, or can it be combined with this yours approach? Thanks.
So the question relates to the technology called deep brain stimulation, where an electrical device is surgically put into the brain of the Parkinson's patient, and then it stimulates certain neurons. I think the fundamental difference between the deep brain stimulation and growth factor therapy is that deep brain stimulation is not keeping neurons alive. They continue to die. Growth factors, the main function is to counteract the death of these neurons. Deep brain stimulation, on the other hand, electrically stimulates neurons, and through that activates biochemical and genetic pathways, and among other things stimulates also the release of certain growth factors. So currently there have been a couple of trials in animals to combine deep brain stimulation with the growth factors, and these look very encouraging. All in all, I think you are doing it also in Tartu. But I think all in all in the world, the number is maybe closer to 300,000 patients who have been treated with a brain stimulation. But not all patients tolerate that well, so it's only to certain. But you better ask that from us, sir. He knows it better than me.
Complex questions have been answered. Any other questions?
Actually, comment is to your question, Jaak. My comment is that if we are talking about how powerful the AI can be, just as a human, psychologists would probably agree that the most important decisions are not made by rational thinking. And similarly, the most important discoveries are not made by rational thinking. So I'm just wondering that the brief maybe is not correct, that we try to make it as powerful as possible if the more important fields are actually just governed by the emotions.
Who said computers have to be rational? Who said that program has to be rational? It can be random, totally random.
There's two things. Mart, you're right. AlphaFold does make a real mess of GDNF. I looked at it, and it'd be interesting to work out why. So you are right that it wouldn't have solved that. On this question about AI, we worked at EBI very closely with DeepMind colleagues to produce the AlphaFold database. It's going to go to at least 100 million proteins coming up, and we hope for every known protein, AlphaFold will attempt to compute a structure. So in your case, GDNF won't be done, but many, many others, and actually away from human, this will have a bigger impact. But what's interesting there is that they did not use generalized AI. They did not write a program that could solve the problem with just the examples or the reality in front of them. They had to write a very, very specific program and cast the problem. There's a lot of programming skill in the development of AlphaFold. And this is speaking to that difference between generalized AI versus very specific AI. So I think AlphaFold is a great example where humans have made a program that can clearly do something that humans don't understand. And that's very, very interesting. But ultimately, humans wrote the program. And Jaak, your question is slightly different. Is there going to be a point where computers can generate the same sort of decision-making, which is very general, that we see in humans, and that's a very different question than the success of AlphaFold, which is remarkable because we don't understand why it works, but it definitely does work. It's sort of orthogonal to this general AI question: can you make a computer which can perform in a way, in a similar way to humans?
But if one neuron receives information, processes it, and transmits, so if we understand the components, in principle, we could be able to replicate that. But the floor is yours, please.
So I think you kind of raised the suspicion why we poor neuroscientists spend time on this GDNF and don't work on big things like
No, absolutely not. No.
But I think that if you really honestly look where neuroscience is today, if you ask me, do I know how memory works in our brain? Honest answer is no. If you ask, do I know how, what is the elementary basis of learning? Do I know? No. So now the question is, if we don't know how biological intelligence works, how can you build based on that artificial intelligence? Not knowing, and you build on not knowing, or what?
Engineering, I guess.
Or what? Or what?
I guess
Or
there
what?
are no
What?
birds who fly like jet planes, so we can engineer different ways achieve kind of similar or more powerful.
So-
But this is philosophical, very
But
philosophical.
science politically, I've been now out from science politics for many years, and I enjoy doing the work in the lab. But I think neuroscience has been in the very bad position. If I look how much money is spent for cancer research and compare that with neuroscience, you can't expect much because the money spent is not relevant.
This could, I mean, obviously, Mart and I may continue with this later on over drinks, but and I actually agree that we disproportionately, potentially, particularly commercially, we disproportionately, for example, favor cancer for all sorts of economic reasons, which is a bit silly. But actually agreeing with your point that we don't understand it, not understanding humans, I think, that's a bold thing. But we can watch a C. elegans brain think. We can hold a C. elegans brain in a vice. Brain, yeah, 600 neurons. We can hold the head in a vice, and we can watch every neuron fire. And we know when we release the worm what the worms do in this situation. And we can watch all the neurons fire. And yet we still cannot come up with a model of how the neurons fire give rise to the decisions made by the worm. And I think that is, so before we do the human brain, I think we should do the worm and the fly brain first in this. But it's going to be a combination. This goes to biology as a data science. We're not going to come up with a theory for all 500,000 synapses of the dopamine neurons in our brains. We will simply have to observe them and use computers, AI and data science, to manipulate this. Much of our understanding will come via computers because we cannot, in our own heads, hold the complexity of these structures. And that's true for protein structures. I'm sure it's true for brains.
I think this is a very optimistic note on the side that there is so much to study for next generation PhD students, postdocs, researchers, getting students into the science. And with this, I think we have to stop this event in here, and we will move out, and we have earned our meals and whatever drinks out there is. Thank you so much, both.